In this video, we are going to go over nucleic acids. So basically, what are nucleic acids? So nucleic acids are large biomolecules that store, transmit, and also express genetic material. While that may be easy to master, we need to understand what genetic material is. So basically, when we are talking about genetic material, we're talking about the hereditary substance, okay, that is stored in the cell. So it's a hereditary substance that is stored in the cell. So basically, when you talk about hereditary information that is stored in the cell, it is basically what carries every information about you. It carries every information about living organisms, meaning that the way a human being may look, the way they act, the way their structure is made up of whatever component of the body, the way it is made, it is because of this hereditary information that is stored in the cell. And that is what we refer to as genetic material. And this basically explains why you may look like your parents, because of the transmission of this genetic material. So nucleic acids are the ones that are responsible for storing that genetic information to help it be transferred from one generation to the other. Okay. So now, when you talk about nucleic acids, we have to understand that basically there are two types of what? There are two types of nucleic acids which differ in functions. So basically, when you talk about nucleic acid, we are saying we have what we call DNA and also what? RNA. So these are very common terms that we've come across. So when you talk about DNA, it actually stands for deoxyribonucleic acid. Then, as you might have guessed, for RNA it's ribonucleic acid. Okay, so both of these are nucleic acids. So they differ in terms of functions. So when you talk about DNA, DNA is more involved with the storage of the genetic information. Then RNA is more involved with the transmission. Okay, and also it helps in expression of the genetic material. Okay, transmission and also expression. Okay, so the structure of DNA allows it to store the genetic information about you, and that's why it's the most famous one, most renowned to people. Okay, so DNA is what stores information about you. Every information that is about you that is stored in a cell, that has information about your color, your height, and everything about you is stored in the cell in the component called DNA. Okay, so DNA only stores. But when it comes to transmission, when there is replication, so RNA is involved in carrying that information to make new cells. Okay, so basically there is only one form of DNA, but when you talk about RNA, there are different forms that we are going to cover in a separate video. So in this video, let's focus more on the structure of DNA and RNA. Okay, so basically there is a single unit of the nucleic acids. So when you talk about components that make up a nucleic acid, we have what we call a nucleotide. So basically, as you may be aware, for example, if you talk about biomolecules, a biomolecule like proteins, it is formed by a combination of amino acids with what? Amino acids combine together to form proteins. So equally, there are some units that combine to form a nucleic acid, and that is called a nucleotide. So a nucleotide has got three major components that make it up its structure. So it has got what we call the phosphate, the pentose sugar, and also the nitrogenous base. Okay. So a pentose sugar is a 5-carbon sugar. Then a nitrogenous base is made up of nitrogen atoms around it, as we'll look at its structure more in details. Okay. So phosphate is it's actually basically phosphoric acid, but most of the times we refer to it as just a phosphate. Okay, so it's phosphoric acid. So all these components are going to be talked about more in details. So the bond that is formed between the pentose sugar and the nitrogenous base is called a glycosidic bond. So that is called a glycosidic bond. And when it comes to the counting of the carbons that are around the pentose sugar, we start from that point. So carbon 1, with carbon 2, with carbon 3, with carbon 4, and there's carbon 5 there. So we're going to talk about these into details one by one. Okay, so basically when you talk about the structure of DNA, this is the way DNA comes out in terms of structure. So since we know now the way a nucleotide comes out, it's a single unit. Okay, so let me write one. So that is a nucleotide. So now when you get to combine a lot of nucleotides together, you get to begin joining them. What you're forming now is what we are calling a nucleic acid. Okay, so DNA, this is the way it comes out in structure. There's base pairing between two different strands that run parallel to each other. Okay, so these bonds that form in between these nitrogenous bases are called hydrogen bonds. So basically the hydrogen bonds are the ones involved in bond formation between the nitrogenous bases. Okay, so on the other hand, when you talk about the structure of RNA, it is only a single strand, meaning that there are no hydrogen bonds between the strands. So it's just a single line or a single strand, which is involving interconnection of what? Interconnection of the nucleotides. Okay, so we've gone back to the nucleotide now, talking about each part more in details. So when you talk about a pentose sugar, what basically is a pentose sugar? So a pentose sugar, this is coming from the word pentagon, because we are talking about a 5 carbon sugar. And when you get to come up with this structure, you're going to see that this is basically the way a pentose sugar gets to come out. So it's 5-sided, a 5-sided shape. Okay, so don't mind the drawing, but that's the way it comes out. Okay, so that is so if you look at the size, is 1, 2, 3, 4, and 5. So that's why it's called a pentose sugar. Okay, so the structure is that there's a hydrogen there, there's a hydrogen there, there's an hydroxide there, there's also an hydroxide there, and at that point there's an hydroxide, a hydrogen there, we have CH2OH with also an hydroxide down there. Then at that point there is oxygen. So that is the basic form of a pentose sugar that we're talking about in this case. Okay, so basically what differentiates DNA and RNA is the fact that when you talk about RNA, what I've drawn is actually a pentose sugar for what? For RNA. So let me try to draw the other one for DNA so that we get to see the difference that is there. And also in terms of the names, how different are they? How do you get to refer them differently in terms of their structures? Okay, so a correction there. So the hydroxide is supposed to be basically on top and not down. Okay, so the hydroxide is on top, then the hydrogen is down there. Okay, so the other one is going to come out this way. So we're supposed to have a 5 carbon sugar, 5-sided sugar. Okay, so there's oxygen there, so there's H here, there's H here, there's the hydrogen there, there's also an hydroxide there. Okay, so basically the difference is only on carbon 2. So the difference comes in when you look at carbon 2. So when you look at carbon 2, remember we said when it comes to the positioning of carbon atoms, we start from that point. So it's 1, 2, 3, 4, and 5. So it's a 5 carbon sugar. So the difference is on carbon 2. When you look at the carbon 2 of the other pentose sugar, there is no oxygen. So due to the lack of oxygen there, it is called deoxyribose. Okay, so the pentose sugar found in DNA is deoxyribose. So the other hand, the pentose sugar this side for RNA is ribose. So these are the two different pentose sugars that make up a structure of a nucleotide, and these are the ones that make the difference between the nucleotide of RNA and DNA to be different, because the phosphate and the nitrogenous bases basically are one and the same. They only come to differ when you talk about thymine and cytosine, and thymine and uracil, which we are going to explain soon. Okay, so apart from pentose sugar, we said a nucleotide is also made up of nitrogenous bases. So these nitrogenous bases are classified into purines and what? And pyrimidines, looking at their link structures. So when you talk about a purine actually is a two-link structure. Then pyrimidines, these are single-link structures. So the general form that we're talking about is something that is coming out like this. Okay, so when you talk about a purine, something like this. Okay. Okay. Then for a pyrimidine, something that is going to come out just like this. Okay. So what is a pyrimidine? So this is what we are calling a double link, and that is a single link. So basically, if you want to remember, you just look at purine, the name is short. Pyrimidines is a bit longer. So the one that is longer has got a single link, then the one that is short has got a double link. So let's get to talk about the different types of purines that we have to start with. Okay. So when you talk about purines, you have to know that there are two types of purines. So we have what we call adenine and what we call guanine. Okay, so guanine. So when you talk about adenine, the structure comes out like this. Okay. Okay, so at that point, I'm going to only highlight the important things that differentiate them from the other ones. So at that point, you find that there's a nitrogen and a hydrogen, which is a point where it gets to connect to the pentose sugar. Okay. Then at that point, what is important is for you guys to note that there is NH2 there. Okay, an amino group. So that is basically the structure of adenine. So the other things, of course, there may be other things. At that point, there is, for example, when you talk about this part, there is a nitrogen there. Okay. There's also a nitrogen at that point. There's nitrogen at that point. Okay. So when it comes to the positioning of the carbons, we start counting from this carbon there. So it's 1, 2, 3, 4, 5, and 6. So then you get to 7, 8, and 9. So we say that the purines or other nitrogenous bases are connected. When you talk about the purines, they are connected at what? At carbon number 9 to the pentose sugar. So remember the structure of a nucleotide. We said there's a pentose sugar that is in between there. Then there's a connection of a nitrogenous base. So at that point, so that is carbon 9 being joined to carbon 1 of pentose sugar. And that bond we said is called what? A glycosidic bond. Okay. So that basically is the structure of adenine. On the other hand, when you talk about guanine, how is it in terms of structure? So I'll draw basically the same link, a double link structure. Okay. So things are just the same. There's only a difference at 3 points. So at that NH, that is where the joining occurs. It's basically the same. Okay, it's also the same at that point. Then the difference is on the carbon 6 that we have at that point. Here there is a double bond to an oxygen, which tells us that there is a carbonyl group at that point. Okay. Then at that point there is NH, and there is an amino group on carbon 2 instead. Okay. So of course at this point there's also a nitrogen. So basically the major difference that you can remember is just looking at that point. So know that at carbon 6 for adenine there's an amino group. Then for guanine there's what? An oxygen. So that would help you to be able to differentiate the two guys and be able to identify which one you are given at each time. So very important. So the key points here, what we are saying is know that the purines get to join to the pentose sugar at carbon 9, and they differ at carbon 6, where there's NH2 for the adenine, then for guanine there's a carbonyl group. Okay, so now we know what purines are, and we're able to identify them and see the differences in terms of the structures. Let's get to look at now the pyrimidines. Okay, so due to lack of enough space, I'll just remove the purines there. Okay. So these are basically 3 and not 2. So we have 3 pyrimidines, but these are single links. So we have basically something coming out like this. Okay. So I'll draw them, then we get to give them different names so that we're able to identify them. Okay, so I'll start with the first part there on top. We see. So for the first one, we're going to have NH2, just like in adenine. Then the second one, just like in guanine. And basically, these last two are going to be the same at that point. Then the other difference is going to be these two, because basically when you look at that point, I've said there is a double bond to oxygen. So how do we get to differentiate them? So the difference is going to be at this point, where one is going to have a methyl group. Okay. So basically that's the major difference. But other things like at that point they are the same. Then these get to join at this point. Okay, this is where they get to form. Let me draw it better here. Okay, just here, so that you're able to see what I'm talking about. Okay, so what I'm saying is this. Okay, so we're saying there's a double bond there for oxygen. So now these, in terms of connecting to the pentose sugar, they connect at this point. So as you might guess, there is also NH there. So that is where the connection occurs. Okay. So basically these are the three types of pyrimidines that we have. Okay, and I hope you'll be able to identify them by just looking at the things that are different there. Because if you look at the first two, there's differences on the 2 points. So this first one is different from the two. So you're able to spot it out and be able to identify it. So this one is called cytosine. So cytosine is more related to adenine. Okay. Cytosine is related to adenine. Okay, because of NH2 at that point. Okay. Then the one with the methyl group is actually thymine. So methyl ends with THY, so that it can help you out to remember. So methyl group, this is called thymine. Okay. Then the one which doesn't have any major difference group is basically uracil. Okay. So each nucleic acid, DNA and RNA, only has got 4 different nitrogenous bases. So cytosine is found in both DNA and RNA. So this one is found in both DNA and what? And RNA. But when you get to talk about thymine is only found in DNA structures. The uracil is only found in what? In RNA structures, as we'll get to understand better of. Okay, so these basically are the different nitrogenous bases that we have and that we can talk about. The other component that makes up a nucleotide that we've talked about is a phosphate, which is coming from phosphoric acid. So this is basically the structure of phosphoric acid. So it's basically joins on carbon 5. So if you look at the structure of the pentose sugar that we had, so we are saying it's on that point where it gets to join. Okay. So basically this is the structure. So there's the 3 hydroxides there. Okay. And the joining occurs at that point. Okay. Okay, so basically how is a nucleotide formed? Since we've already discussed the different components that makes up a nucleotide, so how does the formation occur? So the first thing that occurs in terms of forming a nucleotide is first of all a bond between the pentose sugar and what? And the nitrogenous base. So this is what we are talking about. So we have the pentose sugar there. Okay. So what is the pentose sugar? So depending on what is on carbon 2, whether it's an hydroxide down there or it's just a hydrogen. So if it's an hydroxide, it means that we're talking about RNA nucleotide. Okay, so that's what we have. We start with that as an hydroxide there. So now there's going to be, remember we said here at this point for a pentose sugar, we said there's an hydroxide there and also an hydrogen at that point. So then we talked about a point where the nitrogenous base gets to connect. If you talk about adenine, which was a double link, we said it's on carbon 9 where there's NH. Equally, the same thing applies to the pyrimidines. So it's also NH, there was NH at that point where they are supposed to join. So there was something like this. Yeah. Let me try to draw. Maybe I'll draw guanine. So you say they join at that point where there is NH. Okay. So meaning that this is supposed to bend. So how does it come to bend? So it's going to be like this. Okay, so it's going to bring that part now. Okay, so at that part where there is the nitrogen and the hydrogen, so this part, it is the one that is going to join at that point. So what we have is the carbon 1 for the pentose sugar, and also in this case 1, 2, and the carbon 3 for the pyrimidine that you're talking about. So we are saying in this case what we're talking about is what? Let's say maybe it's let's say it's cytosine, or yeah, cytosine. So I'll put NH2 at that point. So it's NH2 on top, just like adenine. Okay, so in terms of the formation, how does it occur? So there is a reaction there that occurs. Okay, there is loss of water. Okay, so this process is basically said to be what? A condensation reaction, because there's supposed to be a loss of water. So how does the loss of water occur? Between what and what? So it's going to occur between the hydroxide group that is there on the pentose sugar reacting with the hydrogen that is coming from the nitrogenous space. So basically that is what is going to happen there. So what happens when the bond is formed? Okay, so what is going to happen is this. So when the bond is formed, basically what you are going to have is there's going to be a connection there. So this is the way the pentose sugar is going to be. So it is going to be lost there. What is going to be lost is the nitrogen there and the hydroxide. So meaning that on top there will be what? A connection to that structure. So I can remove that weight there so that I show. So there is going to be on top now. What we're going to have is basically the structure of the nitrogenous space. Okay, so that's what you're saying. So basically that's why it's even called a nitrogenous space. So that bond there is called a glycosidic bond. So that occurs in a process called condensation reaction due to a loss of what? Due to loss of water. Okay, so after this connection has occurred, this structure is called a nucleoside. It's not called a nucleotide, it's called a nucleoside. So a nucleoside is a combination of the pentose sugar with what? With a nitrogenous base. So we are saying a nucleoside. So now that you have a nucleoside, the third step that is supposed to occur is now the combination or joining to a phosphate. So a phosphate is supposed to join to that point now. Okay, so what happens when the phosphate, how does it occur in terms of connection? Okay, so we said this is the way the structure of a phosphate is. Okay, we talked about something like this. Let me write it properly. So we have an hydroxide there, and we have a double bond there to the oxygen. Okay, so basically what is going to happen, I said, is this part that is supposed to join. So when it comes to join at that point there, there's also supposed to be a loss of water there. So it's also a condensation reaction that occurs. Okay, so what is going to happen is that we're supposed to have loss of what? We're supposed to have loss of water. So if you look at the carbon 5 of pentose sugar, what did we say? We said we have, at that point, we have CH2OH. That is what we have in terms of structure. So that hydroxide group there is going to be lost and it's going to combine with that hydrogen from there. So basically what you're going to have in terms of a connection is this. So I'll draw it separately. So what you have is this. So at that point what you're going to have is the CH2OH here. What we are losing is what? An hydroxide is being lost. So what is going to remain is the CH2. So we remain with CH2 at that point. Then this is supposed to be connected to what? To the oxygen. So where is the phosphate going? So we are saying at the phosphate there was an hydroxide, so the hydrogen is lost. So it's going to be like that now. So that is the way our phosphate was in terms of structure. So that is what we have as a bond being formed there. Remember, on the other side there is now a nitrogen connecting to what? Connecting to the nitrogenous base. Okay, so what is our structure? So this is a structure of and the formation of a nucleotide. So this all processes. First of all, that one is a glycosidic bond, which is also formed from condensation, and this one now is called a phosphate bond. Okay, a phosphoester bond. So that one is called a phosphoester bond. So basically, that is what you need to know about a nucleotide. So when you look at something that has combined, you're supposed to know how to name the nucleotides as well. So in this case, we are saying if you combine adenine, which is a nitrogenous base, you combine it with a pentose sugar, let's say ribose. So in this case, what you're going to form is what? Is a nucleoside, not a nucleotide, a nucleoside only. So the name is going to be adenosine. That's how it gets to name them. So it will be adenosine. Okay. So adenosine. So when you get to add the phosphate now, phosphoric acid, the phosphate there is going to be what? Adenosine what? Phosphate. Okay. So that is the way you get to name the nucleotides. Okay. So what happens now if you get to have guanine there? So if you put guanine where there's adenine, it's going to be guanosine phosphate. Okay, so that is the way you get to name. If you put cytosine there, it will be cytidine phosphate. Okay, so on the phosphate there, you're supposed to put a prefix representing the number of phosphates that are connected. So when you have a single phosphate, it's monophosphate. If you are double, diphosphate, and this gets us to what we call even adenosine triphosphate. So tri-adenosine triphosphate would represent our ATP that we know of. So basically, when you look at the structure, it's going to be something that is going to be like this. So remember, that is where a phosphate is joined. So another phosphate may join from that point, like that. So when they get to be 3, it's triphosphate. Okay, so basically that is all about nucleic acid introduction. So look out for the next video where we're going to talk about DNA in details and also RNA, so that we get to understand the different forms of these guys that we have. So thank you very much for watching.